Good evening, everyone. Before we begin today's session, I'd like to provide a brief introduction of our speaker, Dr. Parul Varma. An eminent scientist working at the forefront of her field, Dr. Varma is currently a postdoctoral researcher at the University of Texas, San Antonio, where she is working on studying the expression of known and novel SARS-CoV-2 spike protein interactors in the fetal brain and deciphering cell-type specific molecular mechanisms in 3D cerebral organoid models of genetic epilepsies. She received her undergraduate and graduate degrees from the University of Allahabad and the University of Calicut in India, and her PhD from the Center for Cellular and Molecular Biology in Hyderabad. Dr. Varma also has several publications to her name. Her most recent works include in situ nuclear matrix preparation in Drosophila melanogaster and using it to study the components of nuclear architecture. Throughout her academic career, Dr. Varma has received numerous prestigious awards and fellowships, including the American Epilepsy Society's postdoctoral research grant and the CSIR's junior and senior research fellowships. Dr. Varma's vast experience in her field is certain to make this a very enlightening session for us. And with that said, I invite her to address us all.
Thank you, Devika, for such a kind introduction. You're very kind, actually. And thank you, UG STEM Peers organizers, for giving me this opportunity to actually interact with students back in India. And it's really, really nice to get in touch with all of you and to be able to talk to you about some of the things that I really enjoy doing. So while I'm presenting, if you have questions, feel free to stop me. Is that okay, Neha? Or do you want the questions and answers towards the end of the session?
No, ma'am. If it's fine with you, it's fine with us, as long as you're
okay. Perfect. Yeah, because I really want this to be interactive, and I am not, like Neha gave me an idea that most of you are from undergrad, BTech, BSc, MSc level. And if you have if you want to if there are things that you're not able to follow, please stop me and ask me. I would be really happy to have it like a very interactive session going on. So with that, and let me start sharing my screen. 1 second. All right, so everybody can see my full screen, right? Not the presenter's mode?
Yes.
Okay, perfect. Okay, so this is good evening in India. It's actually 6:30 a.m. in Texas over here, very early for a Saturday morning. But let's understand something about brain development and disease that I have been working on. And I hope that this is able to the session is able to see what fascinates me with the brain, and I hope I'm able to spark that curiosity in you as well. So okay, so the human brain is awesome. It functions 24 hours a day from the day you were born, and only stops when you take an exam or fall in love, right? We all have been there. Like the minute we have to do something that is really important, our brain stops working. We forget the answers in our viva. We forget the answers to the question paper. We forget things that we should be remembering, right? But it's not that bad, right? Our brain is awesome, and we really our life is dependent on it. If we can't think, if we can't remember, we can't remember things, if we can't think things, then I don't know how we are going to live, right? So how does it all, so our brain is definitely a wonderful brain, and it weighs around 3 pounds and makes up to 2% of the human's body weight. The human brain contains 86 billion neurons, like that is some number, 86 billion nerve cells. And the two types of prominent brain cells are the glial cells and the neurons. And glial cells are mostly the astrocytes, oligodendrocytes, microglia. All of these cells help in proper functioning of the brain, and they help in proper development of the neurons. They help in the proper functioning. So they are equally important, and they actually outnumber the neurons. So there are more glial cells as compared to the neurons in the brain. And this image that I have put over here is from Allen Brain Institute. And this is just like a periodic table kind of thing that they are trying to make for our brain, like human brain. It's like these are different kinds of neurons that are present. And if you get a chance, you should actually go to their website and see the kind of projects that they do, the kind of the technology and the era that we live in right now. There are so many things possible to explore in a human brain and to understand what's going on in that big chunk of 1.5 kgs of organ that is sitting right on our head, in our heads, right? What is happening over there? What all is in there? And how does it do whatever it does, right? Our memory, our consciousness, our dreams, our thinking, our learning, our forgetting, and our finding things during where there are a lot of things around us, and that's something is called as pattern separation that I will talk about a little bit. So all of these fascinating things are done by our brain, that mass of tissue that is in our skull, and it has diversity of neurons, etc. And it's only now that we are able to like understand those. So, but the question is, where does this all begin, right? It begins with a neural tube formation. So during the development of the fetus, it takes like 9 months for the fetus and the brain to develop. And it starts around like 25 days. And the neural tube is the first structure that forms. And from there, the structure of the brain starts forming, right? And there are special, so like all these 9 months, this organ keeps growing, but it's not just a tissue that is just growing. There are like lots of cells, and there are lots of like molecular mechanisms going on over there that actually help in this development of the brain, right? So to give you a flavor of those things, do not get intimidated by any of these slides, and feel free to stop me and ask me, because I'm not familiar how much neuroscience background you guys have. So please don't be scared of asking anything. So what happens during this embryonic neurodevelopment is that there are these neuroepithelial cells that get converted into radial glial cells. And to simplify it, what is it? It is neural stem cells. So in the developing fetus, in that developing brain, there are neural stem cells that differentiate into this large number of neurons, right? And it is like what we understand as neuroscientists, that during this process, a lot of neurogenesis happens. And gliogenesis that I was talking about, the glial cells like the astrocytes, the microglia, the oligodendrocytes, all of that happen after neurogenesis is complete. So it's a very, very, very tightly controlled process that happens. So there are these, just remember that there are these neural stem cells in a developing fetal brain. And I hope you guys are familiar with the concept of stem cells. They are the cells that can proliferate and renew themselves and differentiate into the kind of, into a different, highly lineage specific cell type, you know? So there are neural stem cells that have the property that they can divide and make another neural stem cell, and then they can also differentiate. They can become a neuron then, right? So for the longest time in neuroscience, people thought that when you are born with a certain number of neurons, that is all you have till you die. So this was a very, very strongly held concept in neuroscience, that you're born with all the neurons you're going to need in your entire life. Once you're born, there are no more neural stem cells. There is nothing that is going to change your brain, and that is how it is going to be. However, it was back in 1955 that there was this scientist, Altman Das, who actually showed for the first time that no, in the adult brain, there are cells that are actually looking like they can divide and they can make new neurons. And those were the cells that he called them as adult neural stem cells, because they were present in a brain that was adult brain. So, but over here, what I'm showing right now is, before I move more into that adult neural stem cell, this is just a molecular landscape of how during development, when the fetus is developing, when the fetal brain is developing, what are the transcription factors that give an identity to a particular cell type, right? So transcription factors, if you all know, that they are just kind of proteins that are present in the cell. And they are the ones that are like, sometimes they're very specific to a particular cell type, right? So neuroepithelial cells will express a factor X or Y, and then radial glial cells will express a factor Z and A, and then intermediate progenitors are going to express factor B and C, something like that, right? You don't have to worry about all of these factors, but all I want to say is that it's very it's like an orchestra. It's like 1,000 instruments playing at the same time. It's that beautiful. It's like it's watching somebody like cooking a meal for like 1,000 people, like doing everything at the same time, you know, like so many things. So it's a very dynamic process where the expression and repression of all of these proteins happen, and they give identity to a particular cell type. So this is what I wanted to highlight about embryonic neurodevelopment, and I will come to this again. So this is what I was talking about, that like for the longest time, people thought, well, you're born with certain neurons and you're going to die with those neurons. There's nothing that you can change. But then it was in 1955 that this person saw, like, no, this is not true. There are neural stem cells, even in an adult brain. And that opened up a whole new lot of possibilities, right? So, but it was not that easy. It took scientists another 35 to 40 years to actually believe what this person was talking back in 1955. So they took a very long time to understand and to prove it. And this is how science works. You have to prove conclusively. You have to show evidence again and again. And only then you can finally conclude that, yes, there are adult neural stem cells or any hypothesis. And then you work on that, like you build upon that concept. So these were the two particular regions in the adult brain that people found that like there are stem cells, neural stem cells in the brain. And this is the ventricular, subventricular zone over here in the lateral ventricles. And this is the dentate gyrus in the hippocampus. So this is actually a section from the mouse brain. And this is the hippocampus. And this is the dentate gyrus. If you can see my arrow over here, my cursor. So, and this is the lateral ventricle. So if you guys, I don't know if you guys saw that news that, well, you all are familiar with the fact that SARS-CoV-2 infects your, it reduces your capacity for smelling, right? And there was a science paper last year that talked about how neural stem cells and neurons in this region actually get infected by SARS-CoV-2, and those are the cells that SARS-CoV-2 attacks, and that's why we have a loss of smell actually. So it's pretty fascinating that these neurons over here and these cell types over here play such a crucial role in our everyday living, right? And I will zoom into these pictures more and help you understand that which region of the brain I am talking about. So see, this is our human brain. This is the temporal lobe over here in pink. And underneath this temporal lobe, this structure called hippocampus is tucked underneath this. And this is a mouse brain section, and this is human brain. And you can see like this dentate gyrus over here. It's like a V-shaped structure. And this is the seat of neurogenesis. This is this region of our brain. Imagine in all our brains sitting right now, there is a structure underneath our temporal lobe, the hippocampus, in which there is this molecular structure, the cellular and molecular structure, dentate gyrus, which is continuously producing new neurons. And we don't even know that, but that is happening. And that is very, very important for our daily living. So this structure is called as the dentate gyrus. And these are the cells present in that like subgranular zone of the dentate gyrus. And again, like I was showing you that during embryonic neurodevelopment, how there are neural stem cells, and then they divide and divide and divide and become a neuron. Exactly like that in the adult brain also, like right now in you and in me, there are neural stem cells in the brain that are dividing and becoming a neuron. So this is where all of that is happening. And the reason it is called as hippocampus, because it's very fascinating. It comes from the it is a sea. In human beings, it's like a seahorse shaped structure. Seahorse is an animal that lives, right? So in the ocean, in the ocean. So it derives its name from that because it looks like that. Excuse me. So this is what I was talking about, that like it is really important for your memory and cognition, for your learning, for forgetting, even for forgetting actually. It is important for your antidepressant. It's important for something called as pattern separation. So, so if you park your scooty, or if you park your car somewhere, if you park your bike somewhere or your cycle somewhere in a parking lot where there are many of those, right? How does your brain know where it is? You know, like how to go, where is the direction? How does it remember all of that? And among so many similar bikes and scooties and everything, I know you're looking at your number plate also, but like, how does it like find out? It's all because of pattern separation. And that pattern separation is guided by the new neurons in our brain, like the ones that are working every day, that are being formed every day. And it's not that like, if all of these are formed, why doesn't our brain grow bigger, right? As we are growing, there are a lot of new neurons that are generated every day, but there are a lot of them that they die. And there are some of them that they integrate in the circuitry, which are actually really important. So it's not that it is producing so many neurons that our brain is going to blow up, but it is like the crucial ones are retained, and then the ones that are not important, like many of them die. And like there is a whole other field of like scientific work over there, like how it happens, which ones are the ones that get integrated into our circuit, etc., etc. But and with this, like when people found out that there are neural stem cells in the brain, so it opened like a whole world of possibilities, you know, like regenerative medicine. So what is that? Like if you had if somebody had a brain injury, if somebody had a stroke. I had this unfortunate incident where my uncle had a stroke, and we know that families suffering from stroke, etc., it is extremely painful. Why? Because there is no cure for that. It says that when you have a stroke, the clock ticks like faster, it's moving faster. And the brain, the region where the stroke hits, it deteriorates so fast that like nothing can be done. You can give them some steroids to help with the inflammation, you can give them certain things, but certain medicines. But if a part of your brain is destroyed, it is destroyed. Similarly, for like Alzheimer's disease, like neurodegeneration, Parkinson's disease, all of these diseases where it is very hard that though we found all of this neural stem cells, that is why scientists are so interested in understanding this, because if you can harness the potential of these neural stem cells, you can treat these disorders. You can guide these neural stem cells with factors, etc., to become the neurons and repopulate the areas that have been damaged by an injury to the brain, right? So that is why this adult neurogenesis is so important. And because you're not like, it's not the embryonic neurodevelopment anymore, this is like the adult brain. And if an adult brain gets an injury, this is fantastic news that there are neural stem cells in the brain, even though they're a small population, right? But despite all of this, there is a bad aspect of this adult neurogenesis. And not a bad aspect, but like when adult neurogenesis, when these neural stem cells become uncontrolled. And that is why in regenerative medicine, it is important that people understand that it has to be a controlled conversion of neural stem cells into neurons and repopulating the damaged area. Because if it is uncontrolled, there can be severe consequences. Imagine if if your neural stem cells were boosted and like you have an explosion of new neurons in the brain, they are going to make, first of all, some of them will die and rest of them are going to make all wrong connections. And that is exactly what happens in epilepsy. So if you all know, epilepsy, as we call in India, is also like fits. So it's when people lose consciousness, their body jerks and shakes and they fall and their muscles get tightened up. And in olden times, people used to say that they are occupied by demons or some evil forces and something bad is happening to them. And they would take them to temples, etc., to like clear off those aspects. But that's not the thing. It's a neurological disorder that is leading to this fit, that is leading to this epilepsy, that is leading to the seizures, right? There are wrong neurological connections. There is something wrong going on in the brain that is making that person seize, having those uncontrollable, the person is shaking and jerking and falling and losing consciousness. So that is what epilepsy is. And when I started working in neuroscience, I started with this neurological disorder, epilepsy. So I'm going to talk a little bit about like how we study this one particular neurological disorder, why neural stem cells are important, how we should use them, etc., etc., just to give you a flavor of like how different neurological diseases are studied in neuroscience, so that you are also like, you get excited about this. Maybe someday you all are going to do a PhD and a postdoctoral and become scientists one day. So to begin with, epilepsy, it's said like 65 million people are there around the world who have epilepsy. 1 in 26 alone in the United States will develop epilepsy at some point of time in their life. And we also have this 1in26.org. It's a website and you can go check it out. Yes, 1 in 26. That is such a huge number. 1 in 26 people can have epilepsy. And there are like 4 and 10 out of 1,000 people like who live with active seizures. Like there no available treatment works for them, for many of them. There are anti-epileptic medicines, yes, in India, in United States, but they are not very effective. People still have seizures. There are kids who have seizures. And it's very heartbreaking and it's very sad to see that. And sometimes we know, like the doctors and the scientists know what is the cause of the seizures, but there are times when even the cause is unknown. And again, that was like the scare, like that people were scared because they didn't know what's happening. And that's why they thought that there are some evil forces occupying this person. And that is why this person is having these fits, right? So cause is not known. They live with active seizures. Sometimes people get 25 seizures in a day. It's like it's basically like they're living a life which is extremely hard, and it is very heartbreaking, right? So we consider that there are 2 aspects to epilepsy. One is that it can be acquired, and another is that it can be genetic. So you can be born with epilepsy. So there are certain genes that get mutated while you are like, there are de novo mutations or in these genes. And then a kid is born, and by 2 years of age, this kid has seizures. And then parents go to the clinic, and then at least in the United States, there is like gene panel testing, and people can diagnose through gene sequencing, like which gene is it. Sometimes they can, sometimes they cannot, which gene is causing this epilepsy. but I'm not familiar that if in India we do this gene panel testing yet to figure out the cause of a particular disease, right? And then another aspect of epilepsy is that it can be acquired. So what happens is like it's unfortunate that if you are in a road accident, or if you hit a brain, or our soldiers who get hit by a bullet or something, they at certain point of time in their life start developing these seizures, this epilepsy. So this type of epilepsy is called as acquired epilepsy, and most of the times this is actually temporal lobe epilepsy. Remember the temporal lobe that I showed in the brain? It starts from there, and I will show you further in further slides that how it starts from hippocampus and what we do about it, right? So epilepsy can either be genetic or it can be acquired. It can be because of the stroke, it can be because of brain tumors, it can be because of traumatic brain injury, it can be because of concussion, right? Football players, many football players, like because they keep getting hit by football on their head, later in life they start having epileptic seizures. So it's a very prevalent disease. It's more than actually we think about, right? We never think that like somebody will start having seizures, and it does not even happen very immediately. It happens after many years actually. So it happens in human beings, it happens after like 10 to 15 years or so after their injury. And that period is called as the latent period. And we do not realize somebody was hit by a football many times during their early growing years, and later in life they start having seizures. So how is that happening? Because all those hits in the brain, they did something to the neurons over there. Everything got messed up over there, and that's why the connections, the circuitry changed, and that's why they're having seizures, right? So this is what I was talking about, that temporal lobe, the mesial temporal lobe epilepsy is a clinical subtype of temporal lobe epilepsy. And this is the temporal lobe again, and this is the hippocampus, and it accounts for almost 60% of all people living with epilepsy. And it begins within a structure of the brain called hippocampus. I already introduced what hippocampus is, and in its surrounding structures, right? And this hippocampal onset accounts for almost 80% of all temporal lobe seizures. The worst part, it is often resistant to medications. And what happens is that no medicines are working. So what doctors do is like they try to find out the region in the brain where they can like pinpoint. I'm sorry if somebody has a question. If a child has epilepsy and experiences seizures since birth, it is curable, or does it inevitably mean that it will persist throughout their life? So so that's exactly what I'm talking about, that we all are working towards. It depends upon what kind of epilepsy, and it also depends upon what medications this person can actually respond to. So it's not that all epilepsy is untreatable. It's definitely not like that. There are lots of cases where people are living seizure-free life with certain medications. That is definitely possible. But again, it will depend upon the kind of epilepsy. If it is a genetic epilepsy that has no cure, then it is difficult. And anecdotally, like it has also been seen that in later years of life, sometimes you have very active seizures in the beginning of your life, and then sometimes they plateau or they die down. There are less seizures later. But yes, again, it will again depend upon the kind of epilepsy that person has. So I hope that satisfies the question. Okay. Why is there such a long latent period for chronic traumatic encephalopathy? Why, I do not know exactly, but I know like a lot of, because it's also the fact that brain is not that fragile, right? It is very resilient. It's not going to just die out in one single incident, or something dramatic is going to happen immediately, until and unless it's a stroke. that completely damages everything. And that is why, like Alzheimer's, Parkinson's, all of these diseases actually take time to develop, right? It's the resilience of the brain that maybe it's fighting back and it's still trying to hold on, but it's like a roof that is like somehow holding on, but then cracks and cracks and cracks and cracks and cracks, and then finally it falls. So we think something like that is happening. Can epilepsy somehow fix itself or go away with age? Again, it's like depends upon the kind of epilepsy. Some people have 2 seizures in their lifetime and then they never have it. Some people live with that. Some people go through the most unfortunate part, which is called as SUDEP, which is sudden unexplained death in epilepsy. People don't know. It's I think their body get completely tired and like they die, but don't quote me on that. People have been working now and there are leads, like there are genetic predispositions to those. So yes, again, like it will depend upon the kind of epilepsy. It's a very variable, as you can see, right? It's very heterogeneous and very variable. All great questions, and feel free to keep asking. Feel free to unmute yourself and let me know instead of chatting also. Yeah. So anyways, so it is often resistant to medication, as I was talking about, and surgical therapy is the best option for these individuals. And one of the classic examples, I don't know if some of you know or if none of you know about this, but there was this person, HM, Henry Molaison, who taught us about memory by losing his. And this is where like people really understood that hippocampus is definitely, as I was saying earlier, alluding to earlier, like hippocampus is important for our daily memory. It is important for our daily learning. It is important for our daily normal physiological activities. So this person, it was back in 1953, this person had a brain surgery because he used to have a lot of seizures. And he was not responding to medications. So a surgeon removed his hippocampus, actually. He had this, and you can look him up, he's on Wikipedia, this whole story, and like a classic neuroscience example, actually. And when surgeons removed hippocampus, he lost his memory. He had no memory at all. And he couldn't make new memories, where like he had short-term memory loss. And later in life, he also started forgetting his early memories. So this was very fascinating, that he remembered things that were before his surgery, but he couldn't make more memories after his surgery. And later in life, he also started forgetting what he remembered from his early childhood or early years before the surgery. And I don't know if you guys have seen this movie, 51st Dates, that Drew Barrymore in that also has this short-term memory loss, and like she cannot remember. She for every single day, it's a new day for her. She forgets about it. Right? So this all goes to our hippocampus. It's if something is wrong in our hippocampus, we are not going to remember things, right? So this is exactly what happened with HM, because his temporal lobe was removed. Of course, he was being treated for seizures, but look at the unfortunate incident that happened, that he could not make any more memories and he didn't remember anything. So this was the person who taught us about memory by losing his. And if you're interested, go read about him. It's a very fascinating story. So again, how does this mesial temporal lobe epilepsy happen? There are multiple factors. So this is what I was talking about, that there is a latent period. There are lots of things that happen in an acquired injury. I'm talking about acquired injury, traumatic brain injury, infections such as encephalitis and meningitis, scarring in the part of the temporal lobe called hippocampus, and blood vessel malformations in the brain, stroke, brain tumors, genetic syndromes, and something called a status epilepticus, which is like having seizures for more than a certain period of time, multiple seizures. So sometimes there's due to in laboratory, we model this using some chemicals. So This is like acute seizures. They happen, and then something happens in the brain, and after that, the person starts having chronic epilepsy. So there are multiple reasons this acquired epilepsy can happen, right? And this acquired epilepsy, the temporal lobe epilepsy, the origins are from the hippocampus. And to bring you back again, that in the hippocampus, this is the region that has got the neural stem cells. This is the region which are forming new neurons every single day and are important for our memory, are important for our learning, and are important for our physiological processes, right? And this is just a cartoon showing, like, in this region, this little blue region, dentate gyrus, all these cells are there. See, it's a tiny region, and how all of these cells are packed together. So there are neural stem cells, and then there are, like, they are differentiating, and they are becoming differentiation, like immature neurons, and then they become mature neurons, right? So this is how neural stem cells differentiate into neurons, and our brain gets that. So an adult brain. So what scientists and doctors saw was that, like, in these mesotemporal lobe epilepsy patients, these, like, the postmortem samples, or once they die, like they surgically remove this hippocampus, they can do some staining. I'm not sure how many of you are familiar with staining, but immunostaining basically means that you can see the cells. What you cannot, if you, like, look at your skin, you just see the color of your skin and the skin, right? But if you put this, like, take a section of it, put it under the microscope, put some antibodies on it, you can actually see the cells in your skin, right? You can see, you all would have seen microscopic slides of various sections, right? Of, like, our cell cycle, like the onion slides, etc., that we study in college, etc. So this is exactly what we do. We take these human brain samples, and they show that how in this epileptic patients, you see, like, in controls, this is so tightly packed and everything. And these are basically neurons, these brown dots that you're looking at. It's not a very high resolution. Of course, you cannot see all the processes over here, but you see how they're dispersed. So it means, like, something something is happening to these neurons. They're not in the place they should be. And that is exactly what happens in epilepsy. When they're not in the place where they should be, they start making wrong connections. They start doing things that they shouldn't be doing. And that's why you start having these fits and seizures. So it was back in 1997 that there was this scientist, Jack Parent, and he showed for the first time in mouse model system that so everybody was like, yes, there is a connection. Something happens with the hippocampus. I know that if you remove hippocampus, then epilepsy, like acquired epilepsy, can be treated. But nobody understood, like, exactly why. What is happening in it? Why is it so important? So this person showed that, like, when this mouse, at least in the mouse model system, when you give them epilepsy, you see, like, there is a blast of all the cells. Like, something happens to these neural stem cells, and they go crazy. They start producing a lot more neurons than they should be making. Like, they have no stop cycle. Like, they are just, like, continuously producing and producing. And he was for the first time showing this, and everybody was like, oh, wow, okay, so maybe that's the reason. So, but it was not until, again, many years later, that we showed that, like, it's actually, if you remove these, like, somehow genetically manipulate these or remove from the mouse brain, the mouse actually gets relieved of the seizures. So this is where I'm going to dive into some of the work from our lab, and then later my work. So what we did, so people started connecting dots, it's like puzzle pieces putting together. So you have people know that if you remove hippocampus, your seizures can be controlled, hopefully. Hippocampus is the seat for adult neurogenesis, that is their adult neural stem cells. It is important. And then this person comes along and puts another layer and shows that, oh, this is what is happening. If this mouse gets seizures, it means that they are having a lot more neural stem cells proliferating into neurons. So there are more neurons. So then next person comes in and says, oh, wait, if I remove these neurons, do these mice will not have seizures anymore? So that is exactly what we tried to do. So this is just a schematic, and this is how we ask questions in science, and this is how we design our experiments. So what we did in the lab was like, there is this mouse, we gave it some chemical, we injected a chemical into it to give it seizures first, because mouse generally do not have epilepsy, right? So we give it seizures. These are transgenic mice, like they have a special gene, which is what this gene is going to do. If I give this mice this drug, which is called as ganciclovir, it is going to deplete or remove the neural stem cells and the the adult neurons from this mice brain. So this mice brain is not going to have any neurons in it. Like the hippocampus, the dentate gyrus is not going to have neurons in it, right? So, and we know that that is important. We know that something is going on with it. So we wanted to see that if this mice, first of all, if it was like a clean slate, it didn't have those, and if you gave them this chemical to induce seizures, and then later, and I said like there is a latent period, right, which takes time to develop. So then we do mice brain surgeries. We put in electrodes, and then we record. We see the seizures. We see that these mice are seizing, and we record like, do they have epilepsy? Are they having seizures? And what we find is that if they don't have these neural stem cells, their chance of like the seizure frequency is actually 40% reduced. So this was like a proof of concept study that yes, first there was a connection, and then yes, they're actually essential. They're important in this. If you remove them, they are not going to have seizures. But did you see that? And let me show you something over here as well, that this is just like a brain section from a mouse, and you see these all brown dots. These are all immature neurons, and you see the animal that has all its neural stem cells and neurons and got seizures. This is a pilocarpine chemical that we give to this mouse that has so many of these, and this is the one that we give the drug to remove all of them, like clear the neural stem cells and neurons. These have like nothing, right? And these are the animals which have reduced epilepsy. So they were like, oh, wow, okay. So if there is a reduction in neural stem cells and these aberrant neurons, because this is not how normal brain of a mouse looks like. This is a lot more compared to what a wild type mouse will look like. So like, okay, if we remove these, they are going to have less seizures. This can be a therapeutic approach. But the problem with this approach is, this was like even before the mouse had his first seizure, his neural stem cells were removed, right? We go to the clinic when we start having seizures. There is like, we can't like wake up one day and decide that we are going to remove our neural stem cells and then we will not have epilepsy, right? We cannot do that. And I'm just checking. Yeah, somebody asked, how do you induce epilepsy in mice? So exactly, that's what. We use a chemical pilocarpine. We inject into the mice, and then 25 or 30 minutes later, they have seizures. Then these seizures, we allow them to seize for 3 hours, and that's what we call as status epilepticus. And then we give them a dose of diazepam, or a medicine that will actually control this, like stop their seizures. And then this is called as giving them acute seizures. They seized for 3 hours and now they don't have any seizures. And then 6 weeks later, that's why we are like recording their seizures through video EEG monitoring. 6 weeks later, so 6 weeks later, they start having recurrent seizures, which is called as spontaneous recurrent seizures. So these mice, if you go near them, they will be like, you can see like very often they'll be like jumping in their cage. They will be having seizures. They will be having fits, right? So is there any other question? No. Okay. So this was great, right? And this is the latent period that I was talking about. And like a lot of things are happening in this. But in reality, nobody is going to get up one morning and say that I'm going to go get my neural stem cells removed, because what if I hit my head somewhere and I develop epilepsy 10 years down the line? So might as well get removed of that. And that is also not feasible, because if you remove your neural stem cells and if you don't have those neurons, your learning and memory and all the other physiological processes are going to get impaired, right? So that's why when I started this work, I asked, like, let mouse have its first seizure. We are going to remove after the mouse had its seizures. We are going to remove all the neural stem cells and its progenies and like all the neurons. After this, like, imagine a patient is coming into a clinic after like first seizure and they come into the clinic. So what is going to happen? I need to develop a method that can actually show me that, yes, if I remove this later in life, like after the first seizure, they can still reduce chronic epilepsy. So I said, like, fine. Earlier they did this ablation studies for like 4 weeks before the seizures. I'm going to, for 4 continuous weeks, for 1 month, I'm going to give a drug to this mouse after it had its acute seizures and see if it does not develop any chronic epilepsy. 6 weeks later, there should not be any difference between the experimental and control mice. I have a question. Won't removing the neural cells altogether have impact on the basic functions? Exactly. That is exactly the point. You've got it. So yes, but this was again a proof of concept studies. And then, like, let me finish this and I will talk about that as well. I don't have slides regarding that, but I will talk about it. So this is again proof of concept, because it's not just about removing them. We are at such an early stage. We are trying to like understand the mechanisms first, and then we are going to use it, like we are going to use techniques that can modulate these. We're not going to remove them. We are going to do something with these neural stem cells or neurons, like, okay, don't fire so much, don't be so active, relax a little bit. We are going, that is called as silencing them. And that's exactly was a follow-up study in our lab. And I'm sorry I'm not talking about it today, but yes, that is exactly the point, because in real life scenario, you cannot remove that. And you need something to just like, you know, like a regulator, like regulate it. So for regarding this, what I saw was that like there was no reduction in seizures actually, and that was like a failed experiment. But in science, there's nothing as a failed experiment, because everything is building upon a concept, right? So I'm like, oh, I gave them drug for like 4 weeks, trying to remove their neural stem cells and neurons, and nothing is happening. So then, like, I thought, like, maybe that is not enough. Maybe so much has gone in this mice brain. Maybe it needs more removal. Maybe something is happening that is like there's still a lot more cells over there. So and this is just a immunohistochemistry again that shows like how, like, in the animals that had this drug, you do not see all these beautiful neurons over here, if you can see these images, right? And I will not get into all of these details. These are the prox1 ectopic granule cells, etc. One thing that I want to highlight over here is that, like, yes, even in the dentate gyrus, you can see like this. Oh, sorry, I'm so sorry. So this is the subgranular zone that we are talking about, which has increased neurogenesis. And this region in between this V, it is called as the hilus. So cells are not supposed to be here. The neurons are not supposed to go here. But in epilepsy, what happens is these cells start going there. And that's why we call them as ectopic neurons. They're not normotropic, excuse me. So they're not normotropic. And this was also one of the conclusions from our study, that if we remove enough of these, all these aberrant neurons, the neurons that shouldn't be where they are right now, if we get rid of them, we can control epilepsy. That was the conclusion, right? And it takes a long time to do that. So this I learned, like, after I said, okay, fine, my 4 weeks of experiment is not giving me what I wanted to see. Maybe it is not working, but maybe this is not what is going to help in reducing chronic epilepsy. But let me try doing it for a longer period of time. Maybe the whole physiology is different after the acute seizures, after this mice already had seizures for 3 hours. So I went ahead and like gave the drug, and it's with these little pumps that we put in the back of this mouse, and they keep giving the drug in it. And so I did that, and lo and behold, I saw there was a 65% reduction in chronic epilepsy in these mice at the same time point that I recorded earlier. So, but if they're seizing for like 30 seconds, even the mice which had less seizures, they also seized for 30 seconds. And that's what like this graph shows over here, SRS duration. Like it was not that the control mice seized for like 1 minute and the ablated mice seized for 30 seconds. It was not that. It was like the same amount of time they were having that fit. But SRS frequency, the number of seizures that this mouse had every single day was dramatically reduced from the mouse that was actually did not receive this drug and had its neural stem cells and neurons intact in the dentate gyrus. So again, this is just a histology. We call it as histology. These are the beautiful dentate gyrus from mice, which show like how these neurons look like. And we again see like there is a huge reduction of all of these cells in these mice, so which again concludes that like, yes, if I ablated for a long time, there was a reduction. And we call this as aberrant neurogenesis because this is not normal, right? This explosion of neurogenesis is not normal. So we did that. And then I was very excited and I wanted to see, okay, so imagine a person comes in a clinic after his first seizure, he's treated for 2 months. He should not have to come back to the clinic again. He should be all good, right? Like that medicine should work, everything should be fixed and like, great. And so it means that like the effect of the medicine should last longer. And this is what I was trying to do over here. And I waited, like I ablated for 8 weeks. I know like at this point of time, they are going to have less seizures, which is great. And I wanted to see if this phenomena persisted for a long time. So I waited for almost like after 8 weeks to 18, like 10 weeks, which was like almost close to 3 months. And then I recorded again from these mice. I did the video EEG monitoring and I found that, well, their seizures were back. So it was like not back exactly, but it was like, there was not very, I remember somebody was asking like, do they die out seizures? So see I don't know if you noticed, but like even the control mice started having, like did not have a lot of seizures, but how many ever control mice had that? The ablated mice also had those many seizures. So for sure, this was working for a short time, but it was not working for a very long time actually. So, and that's when we dived into another aspect where we are trying to modulate neurogenesis in a certain way without doing this like hammer approach, like, you know, like clean everything out. No, let's pick and choose. Let's try to see like a regulator. Let's try to see if we can tune it down a little bit, or tune it up a little bit and see how we can have long lasting impacts, right? So to summarize this part of my talk, like this was just a model, like you see like with status epilepticus or acute seizures, there's so many seizures and there's all these neurons are going crazy. You do a 4 week of ablation, there's some reduction, the SRS is still there. You do more than 4 weeks of ablation, there is, yes, you see a reduction, but later there is like, again, there's no difference between the control and the transgenic mice. So it was like maybe other cell types are involved, maybe it is some other inputs that is coming into the dentate gyrus, or something more is happening, and that's where we started exploring more. But with this, all great, but this is all mouse studies, right? This is all mice. Mice brains are not mini human brains, right? We all can appreciate that. And this is just an image which shows like human, macaque, and mouse, and you can appreciate the difference. Look at the tiny brain of mice. So no matter how much, they are great. We have learned so much from mouse model systems, but we are not going to recapitulate what happens in a real human brain if we keep studying mice brain. The reason we are not going to do that is because there are remarkable differences between a mouse and a human brain. And I hope you can appreciate in this picture. So first of all, our cortex is so expanded, right? Look at this mouse. It's like tiny blip somewhere, right? Why is it like this? We scientists think that this, if you can see my cursor, there is a zone, outer subventricular zone, that has got a lot of specialized neural stem cells that are not even present in mouse. They're called as outer radial glial cells. And so they think that this big cortex that we formed in evolution, and that this big thing that happened, a lot of it, they owe it to the presence of this outer subventricular zone, and it's not there in mouse, right? So there are critical differences between a mouse and a human brain, and we need systems that can actually help in understanding neurodevelopmental disorders in human specific neurodevelopmental disorders, right? So Neha, how much time do I have?
It's 7 p.m. So maybe about 5 minutes or so more if you could.
5 minutes or so? Okay, great.
Yeah.
Yeah, I should be able to finish. I really wanted to, because we started a bit late also, right?
Yes, exactly.
So,
That's okay. So
okay, because this is something very exciting and I want to share with everybody. So,
yes,
so
no
this,
worries.
so that's why we need models which can truly mimic neurodevelopmental disorders in human, right? And right now, the field where it is moving is, this is just a cartoon showing that what we can do. We are living such exciting times of science. What we can do, we can take blood from a person or fibroblast, like the muscle, something from the muscle tissue, convert them into iPSC, induced pluripotent stem cells. We can differentiate them into neurons, or we can differentiate into 3D structures, which I will talk about in the next slide. And we can do a high throughput drug screening. We can do so many things. And it's really like from bedside to bench to back to bedside. The concept that we have had for many, many years, it's now that we are able to like do this, right? So in our lab also, we do this. We have patients with genetic mutation, and I'm not talking about that work yet because it's unpublished work. And we got blood from those patients. We converted them into stem cells. We are growing them into something called as brain organoids. These are like 3D cerebral structures that we call this as mini brains in a petri dish. And it's like these are induced pluripotent stem cells, and they differentiate them into immature cerebral organoids, and like they let them grow in the petri dish for like 4 to 6 months, and it has all this, like, it is not all the structures, but it closely mimics the structural features of a human brain. Actually, this is so exciting. We never thought that we would be able to do this in a petri dish, in a tissue culture lab, growing a brain, a mini brain in a petri dish. So this is just to give you an idea. This is how it looks like if you take a section of it and you do an immunostaining. These are the neural stem cells. All the blue that you're seeing, they are the nuclei of the cells, and all the green that you're seeing, these are the neurons, and these are all the neural stem cells in this. And what was the basis? Sorry, I missed this question. Okay, I will come back to these questions. The blood-brain barrier. Yeah, exactly. So this drug, it does get into the brain. It ablates the neural stem cells. Actually, it crosses. Then what was the basis for selecting this drug at the very first moment? Okay, I will get back to that. The reason was that this mice if you remember, I kept saying that there was nestin TK mice. This is thymidine kinase, and GCV actually phosphorylates this thymidine kinase, and this thymidine kinase, then phosphorylated thymidine kinase, kills the cells. That is why we use this drug. So this drug specifically does something to the enzyme present in this mice, and it's a transgenic mice, and then it will kill the cells wherever it is present. So, and it specifically kills the neural stem cells, actually. So I hope that answers all the questions. Yeah. So then these are the neural stem cells in the brain organoids in a dish, right? And this is all we can do with it. Like, we can study congenital brain malformations, major psychiatric disorders, neurodegenerative diseases. We can do drug screening. We can do human evolution. We can understand what makes us human. Why human brain is human brain and not a monkey brain or a mouse brain. What are the key differences? These we can do it now. right? So one example I will give you is like we all have been living in this era of SARS-CoV-2, right? Scientists were able to use human brain organoids to understand that how SARS-CoV-2 affects, which cell types is it going to affect. And they showed that there are like these neurons are there, SARS-CoV-2 can affect them. So this was very closely mimicking the post-mortem brains, right? And this is how they do it. They just take stem cells, they expand them, they dissociate them, they make these organoids, they infect with SARS-CoV-2. And when they infect with SARS-CoV-2 and later they try to see where is it localizing, where is it going in the brain. And this is just an image showing that it is, and I know this, if you're not familiar with immunostaining, this can seem a little bit daunting, but what is this picture is showing that SARS-CoV-2 affects the astrocytes. The astrocytes, which are such important cell types in the brain, they're really important for like proper pruning and everything of the neurons. So these, the glial cells that I was talking about, right? So they have a marker called GFAP. So SARS-CoV-2 affects them. They showed this, right? So what next? So we have this great tool of organoids and everything, and we have learned so much from our mouse model systems. We have learned so much from I have another question. Sorry, can brain organoids be transplanted in a mice to study the behavioral changes in response to ablation medication? Fantastic questions. You guys are so awesome. So, so brain organoids, people are trying to do that. So brain organoids, the lacking feature, which I'm not talking about today, is like they don't have blood vessels. And it's only now that brain organoids, what people do, like after they have grown them for a certain time, they transplant them into mice brain. And when they transplant them into mice brain, the blood vessels start innervating them and they become more mature. How, as exciting as these brain organoids are, they're not like real adult brains, right? We are more mimicking them as fetal brains. So yes, we are trying to develop those technologies. There are a lot of scientists working on them. And that's the goal. Like someday, like if that's possible, one of my colleagues, he's working on using these organoid models to, like, if there's traumatic brain injury, can you transplant these organoids into them and see if there can be a repopulation of the neurons, right? So yes, those are the kind of things we are interested in doing. So what next in neuroscience? And I really want, if you're interested in neuroscience and passionate about it, you should go check out these websites, the Human Connectome Project, like how people all over the world, like the scientists are coming together to map the connection among all of these neurons, right, in the brain. And you should, again, I will say that check out Allen Institute website and see their MindScope program, investigating how computations in the brain give rise to behavior and consciousness. And next is like they are trying to, there are 86 billion neurons, and they have taken up the responsibility that we are going to identify all of these neurons. And they have started with mice, they've started with monkey, they've started with human, and like they are publishing papers. If you're interested, reach out to me, we can talk about it. All the various kinds of neurons and how they are similar and different between a mice and a monkey and a human, right? And I didn't have time to talk about this, but I want to emphasize that with this explosion of data in neuroscience and the technologies where we can like tease apart every single cell and understand about them, has led to a lot of like molecular people like me to start acquiring bioinformatic skills, right? I was a hardcore trained chromatin biology molecular person, and now I do bioinformatics, I do coding, I try to understand, make sense of the single cell RNA-seq data. So if you all have an opportunity, I would say like sit down, take time to learn and code. You don't have to be a computational software engineer to learn to code. If you have this skill, neuroscience, I'm sorry. Neuroscience is at a stage where we require people who understand molecular cellular approaches and can do bioinformatics as well. And with this, thank you so much for your attention. And I am very thankful for my mentors, my lab mates, my collaborators, my funding sources, everybody. And I'm very thankful to UG STEM peers actually for giving me this opportunity to interact with you all. And it's exciting, and I'm happy to take more questions. Let me stop sharing, and I have some questions in the chat, and I will come to that. So is there any way that damaged neurons be reversed or treated except for generating new neurons, as you mentioned? So as far as I understand, I am not familiar with approaches where you can like reverse the damage of the neuron. Once it is dead, it is dead. You can stop from like further death spreading into nearby regions, but when it's gone, it's gone. So, and then next question is, you spoke about brain organoids that you developed in petri dishes using tissue engineering and stem cell technology. I was wondering if similar techniques are being used in other areas to get a better idea of the human body. Absolutely. A lot of organs, like people are growing lung organoids, they are growing pancreatic organoids, they are growing, not familiar with heart organoids, but yeah. So they are liver organoids, and like to closely mimic what goes on in a human body rather than in a mouse actually. So yes, they are doing all of that. Is there any other question?
I don't think there are any more questions unless you received any via direct message.
No, I don't think so.
Okay, well then in that case, if there are no more questions, then I'd like to call on Juhi from the UG STEM peers team to deliver the vote of thanks.
Hello. So a very good evening to all those present here. I consider it my privilege to be proposing a vote of thanks on this occasion. So on behalf of all my fellow peers at SciExplore, and in fact, the entire organization of UG STEM peers, along with all the science enthusiasts present today, I would like to express my heartfelt gratitude to Dr. Parul Varma for her most elucidating discussion on unlocking the mysteries of brain. So I must say, I think what struck me the most was when you explained continued adult neurogenesis in the dentate gyrus, and then further its scope in regenerative medicine, ma'am. And I'm also sure that this talk has resonated with multiple people present today here as well. So thank you for taking out the time for us today.
My pleasure, of course. Thank you so much. I hope you guys were I was able to spark that curiosity, and like some of you will go ahead and make careers in neuroscience. Okay.
It was a really enlightening session, and as Juhi said, thank you so much for agreeing to do this with us.
You're most welcome. Most welcome. Somebody is asking for my email address, and so Neha, if you want, you can share my email address, my Gmail, and if anybody wants to reach out to me, please feel free to do that. I was an undergrad not many years ago, so I totally understand the anxieties and everything that we go through making a career and everything. So I'll be happy to help in any possible way.
Sure, ma'am. I'll send out your email address with a small feedback form to the people who attended the webinar, so that if any of them have any questions, maybe about what you discussed today and didn't want to ask her, I'm sure they'd be very keen on speaking to you about the topic if they have any questions.
Sure, my pleasure.
Thank you so much.
Okay.